1.2, a typical stamen is shown in the A part. You can see the filament or the stalk, and you can see the anther region, which is here. Okay, so this is the anther and this is the filament. And in the B part, you can see the three-dimensional cut section of anther. So here you can see the pollen grains which are there, and you can see the pollen sac. Okay, you can see the line of dehiscence on either sides, and this is again the filament. So this is the anther region which was cut. Okay, so when you cut this anther like this, and when you view it, you can view it like this. You can see the pollen sac and pollen grains along with the line of dehiscence and the filament or the stalk. This is figure 1.3, a transverse section of a young anther. If you can see, this is the first figure. So in this, you can see the outermost epidermis, the second layer endothecium. Then you can see the sporogenous tissue, the innermost, which is coming inside the tapetum. And then you can see the middle layers here. So it's actually epidermis, endothecium, middle layers, tapetum, and then inside that you can see sporogenous tissue. And then here you can see a single round structure that is a connective. Now in B part of this, it is told that it is the enlarged view of one microsporangium showing wall layers. So if you see here, this is just a part which is being shown in large way, that is in a microscopic way. So here you can see the epidermis, endothelium, middle layers, microspore mother cells are shown here. Then you can see the tapetum, and that's all in that case. That is B part. C part shows a mature dehisced anther. So this is how it is, and you can see the mature dehisced anther with pollen grains here. So this is how the structure is. It is almost butterfly shaped structure. This is figure 1.4, scanning electron micrographs of a few pollen grains. As we already know, the variety of architecture, that is sizes, shapes, colors and designs seen in pollen from different species is being shown here. So you can see how the pollen grains actually look. They are different, they look different, their size, the shape, the color, the design, everything look different. There are a lot of varieties that is being mentioned in this particular figure. Now this is figure 1.5, you can see the enlarged view of pollen grain tetrad. So you can see it is a tetrad, right? It's divided into 4, so it's a tetrad. Now you can see the stages of microspore maturing into a pollen grain. So you can see the different stages, a microspore maturing into a pollen grain. So this is where it begins. A single cell, vacuoles are there, nucleus is there. Asymmetric spindle formation happens. There forms two cells, that is vegetative cell and generative cell. So when pollen grain is mature, it contains two cells, the vegetative cell and the generative cell. So we know that the vegetative cell is bigger, has abundant food reserve and large irregularly shaped nucleus. Generative cell is small and it floats in the cytoplasm of the vegetative cell. It is spindle shaped with dense cytoplasm and a nucleus. Over 60% of the total angiosperms, pollen grains are shed at this particular two cell stage. So pollen grains are shed at this particular two cell stage for almost 60% of the total angiosperms present. Pollen products shown in figure 1.6. So this is like pollen product. This is a cream which is being shown, energy shake shown. Then you can see the pollen flower extract shown there. Now this is bee pollen from England. This is also a natural food from honeybees, something like a 30 tablet. That means it's just a tablet or something. But these are the pollen products. You have a lot of products. You can use it as food supplements also, these pollen tablets. So they are really useful, and different ways you can use this pollen. Now this is figure 1.7, a dissected flower of hibiscus showing pistil. So you can see the pistil here, that is stigma, style and ovary, right? So you can see how many 1, 2, 3, 4, 5 here, okay, and then you can see ovary, and then you can see thalamus. This is A part. B part shows a multicapillary syncarpous pistil of papaver. You can see the stigma there, and then here you can see the different carpels. In the third part, where it is multicapillary apocarpus, gynoecium of Michelia. Now the last one is very important, a diagrammatic view of the typical anatropous ovule. So here if you can see the funicle then reaching hilum, okay, hilum, and then it just enters, okay. So through this it enters, right? Alright, so what just happened here? This is the funicle, then you can see the hilum here, then this is like two layers are there, that is outer integument and inner integument, and inside here you can see the nucellus. This is the embryo sac, this is the chalassal pole, while this particular opening is micropyle and this particular pole is the micropylar pole. This is the chalassal pole, this is the micropylar pole, this pole has a micropyle there, yes. So you can see hilum is there, funicle, micropylar pole, outer integument, inner integument, nucellus, embryo sac, chalassal pole, everything is mentioned, that's it. This is figure 1.8, parts of ovule showing large megaspore mother cell, dyad and tetrad of megaspores. So if you can see the micropylar end, the nucellus and the megaspore mother cell. Here also you can see the micropylar end, the nucellus, and you can see megaspore dyad, two of them, 1, 2. And in this case you can see a tetrad, that is 1, 2, 3, 4. Yes or no? That's the first part. The second part is 2, 4, 8 nucleate stages of embryo sac and mature embryo sac. That is B part. 2, 4, 8 stages, yeah, of embryo sac and a mature embryo sac is shown at the end. So this is the mature embryo sac. So here you can see the 3 antipodals, then you can see 2 polar nuclei, a central cell, this is a central cell, egg cell is there, 2 synergids are there, egg cell plus the synergid is called as egg apparatus, okay. Now when you come to the end, you can see a diagrammatic representation of a mature embryo in this particular figure. Chalassal end is where you can see the antipodals, and micropylar end is there where you can see the filiform apparatus here, and then you can see the synergids, the egg cell, then the central cell with the 2 polar nuclei. So it's a 7 cell condition, am I correct? Or is it 8 cell? Let's see, you can have 1, 2, 3, 4, 5, 6, 7 cell, 8 nucleated condition. This is very important, okay. 8 nucleated, 7 cell condition, very important to be noted. So when you talk about nucleated, you always talk about 1, 2, 3, 4, 5, 6, 7 and 8. So like this you have 8 nucleus, so 8, but 7 cell, because the central cell contains 2 polar nuclei, and this 2 polar nuclei can be taken as one. Then the central cell, the remaining each one of them are each cell itself. So therefore 7 cell but 8 nucleated, okay, because one cell, that is a central cell, contains 2 nucleus. This is like figure 1.9 showing A, B and C as self-pollinated. This is self, okay, and then this is cross-pollinated, and then in the last case you can see chlasmogamous flowers. Actually here you can see chlasmogamous closed and chlasmogamous open flowers. These are very important, okay. So make sure you learn this particular figure. Cleistogamous and Chasmogamous. This is a figure which is 1.10 showing the wind pollinated plant showing compact inflorescence and well exposed stamen. So you can see the well exposed stamen here. This is figure 1.11 where you can see pollination by wind in Vallisneria here. The female flower, the stigma is being shown, the male flower is being shown here, and the female flower is being shown. That is how it is getting transferred, is being shown here. That is pollination by water in Vallisneria. The second is insect pollination. You can see a fly here, and this is insect pollination. Now this is figure 1.12. Pollen grains germinating on the stigma is shown in figure A, and pollen tubes growing through the style is shown in B. The LS of pistil showing a path of pollen tube growth is shown in C. So in C you can see the pollen tube, the antipodal cells, the polar nuclei, the two of them, then the egg cell, the synergid, and how the pollen tube is entering, right? Entering. So that's longitudinal section of a flower showing the growth of pollen tube. Okay, and now we have D. D is actually the one which shows an enlarged view of egg apparatus showing the entry of pollen tube into a synergid. So you can see this is how it is. Plasma membrane is there, the synergid is there, the X nucleus is there, inside the egg cell obviously, the central cell, polar nuclei is there, filiform apparatus, male gametes entering the vegetative nucleus. And here you can see discharge of male gamete into the synergid and the movement of sperm, one into the egg and the other into the central cell. So you can see two of them are given, the male gametes, okay, and one is entering into the egg cell while the other is going towards the polar nuclei. This makes it 3n and this makes this 2n. This is why it is called a double fertilization. Yes or no? Yes, that is a discharge of male gamete into the synergid. So here you can see the synergid and the movement of sperm, one into the egg and the other into the central cell. What exactly is in figure 1.13? Fertilized embryo sac showing zygote and primary endosperm nucleus, that is PEN. So you can see the degenerating synergids here, and then you can see the zygote, then you can see the PEC, that is primary endosperm cell as a whole. Then you can see the primary endosperm nucleus, which is in a 3n condition. I've already mentioned how it is 3n, that is 2n plus n. And then you can see the degenerating antipodal cells. So all this together is in the part A. Fertilized embryo sac showing zygote and primary endosperm nucleus. Second case is stages in embryo development in a dicote shown in reduced size as compared to A. Okay, you can see the zygote. Next stage, later you can see the globular embryo, the heart-shaped embryo, you can see the suspensor being developed there, okay, then you can see the plumule and the cotyledon here with the radicle. So you have radicle, cotyledon, plumule. This shows the mature one, right? The others are just the different stages, the developing, and finally it is mature. The last one shows the mature one, okay, as it's written here. Figure 1.14, a typical dicote embryo is being shown in A part. You can see the root cap, the radicle, the hypocotyl, the cotyledons, and the plumule. This is a typical dicote embryo. Now, LS of an embryo of grass. Grass is monocote. So monocote is being shown here. You can see the scutellum, okay, and the coleoptile. The shoot apex is being shown here. The epiblast, the radicle, the root cap, and the coleorhiza as a whole. Coleoptile, coleorhiza, okay, and then you can see the radicle, shoot apex, epiblast, root cap, scutellum. Scutellum is the cotyledon, yeah. See, in grass family, a cotyledon is called scutellum, that is situated towards one side lateral of the embryonal axis. Now this is figure 1.15, structure of some seeds and false fruits of apple and strawberry. Structure of some seeds have been shown in A part. All this is A. So you can see cotyledons, micropyle, the seed coat, the endosperm, the cotyledon. Then you can see the hypocotyl root axis, then the shoot apical meristem, the root tip. And here you can see the endosperm, the root tip is being mentioned here, the hypocotyl root axis, the shoot apical meristem, the cotyledon, the seed coat. Here you can see the pericarp, the endosperm inside, the scutellum here, the coleoptile, the plumule, the radicle, and the coleorhiza. Now you can see false fruits of apple and strawberry. So this is apple. Apple has a seed. It has a thalamus. Now when you talk about the seed, this is the seed, yeah, and you can see the endocarp, the mesocarp. Okay, so all these are there in this case. Now this is the thalamus again. Here is the thalamus, and this is the achene. Okay, the achene. Summary: flowers are a seat of sexual reproduction in angiosperm. In flower, androecium consisting of stamen represents the male reproductive organs, and gynoecium consisting of pistils or carpels represent the female reproductive organ. A typical anther is bilobed, dithecous, tetrasporangiate. The pollen grains develop inside the microsporangia. 4 wall layers are there: the epidermis, endothecium, middle layer, and the tapetum, which surround the microsporangium. Cells of sporogenous tissue lying in the center of the microsporangium undergo meiosis, microsporogenesis, to form tetrads of microspores. Individual microspores mature into pollen grains. Pollen grains represent the male gametophytic generation. The pollen grains have 2 layered wall, the outer exine and the inner intine. Exine is made up of sporopollenin and has germ pores. Pollen grains may have 2 cells, vegetative cell and generative cell, or 3 cells, vegetative cell and the 2 male gametes at the time of shedding. And we already learned 60% of them will be shedding at the stage wherein it is 2 cell or in a 2 cell stage condition. The pistil has 3 parts: stigma, style, ovary. Ovules are present in the ovary. Ovules are present in the ovary. Now ovule have a stalk called as funicle, a protective integuments are there, and you have an opening which is called as micropyle. The central tissue is the nucellus, in which the archisporium differentiates. A cell of archisporium, megaspore mother cell, divides meiotically, and one of the megaspores form the embryo sac or the female gametophyte. The mature embryo sac is 7-celled and 8 nucleate, very important. At the micropylar end is the egg apparatus consisting of the synergids and the egg cell. At the chelasel end there are 3 antipodals. And at the center there's a large central cell with 2 polar nuclei. The pollination is a mechanism of transfer of pollen from the anther to the stigma. It can be from the same plant, different plant, one flower, different flower. All those differ into different categories. Pollinating agents are either abiotic in case of wind and water, or biotic in the sense animals. Pollen pistil interaction involves all events of the landing of pollen grains on the stigma until the pollen tube enters the embryo sac when the pollen is compatible, or pollen inhibition when the pollen is incompatible. The following compatible pollination pollen grains germinates on the stigma, and the resulting pollen tube growth through the style under the ovule. finally discharges the two male gametes in one of the synergid. Angiosperm exhibit double fertilization because two fusion events occur in the embryo sac, namely syngamy and triple fusion. The products of these fusion are diploid zygote and a triploid primary endosperm nucleus. PEN in the primary endosperm cell. Zygote develops into embryo and primary endosperm cell forms the endosperm tissue. Formation of endosperms always precedes the development of the embryo, and developing embryo passes through different stages such as proembryo, globular, heart-shaped, all those stages before maturation. Mature dicotyledonous embryo has two cotyledons. Monocots have one cotyledon. Here it has two cotyledon and an embryonal axis with epicotyl and hypocotyl, four dicotyledonous. Embryos of monocotyledonous have single cotyledon. After fertilization, ovary develops into fruit and ovule develops into seed. Fruit is always known as the ripened ovary, okay, or mature ovary. Now, phenomena called apomixis is found in some angiosperms, particularly in grasses. It results in the formation of in the formation of seeds without fertilization. Apomix have several advantages in horticulture and agriculture. Some angiosperms produce more than one embryo in their seed. This phenomenon is called polyembryony.